hear me? All right. As most of you know, I'm Sonia Hurt, and I'm the Dean of the College of Environment and Design, and I want to welcome you to yet another wonderful presentation that is part of our faculty lecture series. It is my privilege to introduce you to Jenny, get all the two names together right, Cruse-Sanders, who, as you have read and it says on the slide, is the director of the State Botanical Garden. She's here among friends. I'm extremely pleased to say that from what I've seen, we don't only have faculty from CED and students from CED, but also some from the all-important Department of Horticulture, which I think is very dear to your heart. There is a small error that I committed inviting her. I didn't think it through, and this week is actually the conference, the annual conference of the Society for Horticulture Science, which explains why more of our horticulture faculty could not be here. But we also have some colleagues here and students from forestry and other schools, which is of course wonderful because partially the purpose of these lectures is to increase the visibility of our college and to encourage interdisciplinary discourse. So again, it is a great privilege to introduce Dr. Cruse-Sanders. And by the way, the Botanical Garden, in case you didn't know, is celebrating its 56th anniversary this year, which I think makes it exactly equal in age to our school.
Wow.
Did you know?
I didn't know we shared that.
We were founded in 1969. It was the summer of 69.
Okay.
Is that the same year?
We were 68, but it was a process.
They predate us by one full year. Oh, sorrow. Anyway, so in case you haven't been, how many people have not been to the Botanical Garden?
Wonderful. Okay.
Very few. You must immediately correct this. So the State Botanical Garden is a 323-acre university garden with 33 acres of cultivated gardens and more than 5 miles of trails through significant natural areas. It is part of the public service and outreach operation at the University of Georgia, and it serves the communities of Georgia and beyond through educational programming, horticultural expertise, display gardens, and conservational programs. Dr. Cruse-Sanders has an MS and PhD in botany from the University of Georgia, and I do hope we have some people from this department as well. And she has completed her BA in biology at Boston University. She has an extremely distinguished biography. She is the recipient of the 2016 Marsh Award for International Plant Conservation from Botanic Garden Conservation International, the 2016 Carl Becker Stewardship Award from the Natural Areas Association, and the 2015 USDA Forest Service Wings Across America International Award for Urban Communities in Conservation. Through collaborative partnerships, she has helped to build networks for conservation across the Southeastern United States and to develop community sustainability programs to establish native plants and pollinator habitats in green spaces. So as far as I understand, some of the work you're presenting is connected to an NSF grant.
Yes.
That you...
and I will refer to that.
And this is absolutely wonderful. We, of course, encourage our own faculty to participate in interdisciplinary submissions. So the career of Dr. Cruse-Sanders in this regard is absolutely exemplary. So perhaps that will bring more collaboration. So again, let us welcome her. Thank you so much for coming. A round of applause, and then I'll decide. Thank you.
Thank you, Sonia. And I truly appreciate the opportunity to be here. I'm delighted to be here and talking with you. And I see friends and colleagues around the room, and I'm pleased to talk with you about conservation horticulture. And part of this presentation is one that I gave at Smithsonian Institution at their annual botany symposium earlier this year. And why that's notable is because there's emerging interest in conservation horticulture globally and how we can contribute to conservation. I'll start with the photo, which is Eriocolon corniciatum, which is a G2 or globally imperiled or vulnerable species, which is vulnerable to extinction throughout its range. Here it's being propagated clonally at the State Botanical Garden of Georgia. So it's just one example that we'll see today. So our question, the question we ask at the State Botanical Garden of Georgia among my colleagues is, how can we translate what we know about how plants grow to better conserve them? This is important because the latest reports, including the 2023 Kew report on the state of the world's plant and fungi, documents that 45%, nearly half of the flowering plants around the world, are threatened with extinction. 77% of undescribed plants, plants that haven't yet been described scientifically, are threatened with extinction. So this is a picture of Trillium georgianum. It was named in 2017. It's an example of one of those species that was just recently named scientifically, just recently determined to be its own unique species. It's a G1, meaning it's critically imperiled, extremely vulnerable to extinction, and it is propagated and safeguarded. These are plants that are safeguarded at the State Botanical Garden. It's endemic to a single location in Georgia, one spot in Georgia, and it could so easily blink out of the landscape. But with conservation horticulture, we're propagating it and safeguarding it. It's one of the tools in the toolbox. A report by Knapp et al. Recently published in 2021 found that 65 taxa have gone extinct in North America since the mid-16th century. That's an extinction rate of 1.4 species per decade. Now, it's not a plant, but if you're listening to the news this week, we've just recently, in Science, identified a new, what I would characterize as a charismatic megafauna. A new species of ghost shark has just been scientifically described. That's not something little. That's not a bacterium or a fungus. It's a shark. How did we miss that? So there are species out there that we're just beginning to discover and understand, while most of biodiversity, or potentially nearly 50% of biodiversity, is disappearing before our very eyes. We have a significant role to play in helping prevent that. What's also really, really interesting to me is that not just the loss of species, but as they begin to disappear, and research reports have been coming out over the past 15 to 20 years that have modeled species extinctions and found that even as species become rare, their function in the ecosystem is lost. Their connections with their community are broken. And what's very interesting to me is that rare species have been found to disproportionately increase the potential breadth of functions provided by ecosystems across many spatial scales. And they are likely to ensure against future uncertainty arising from ecosystem change. So for example, 2 examples, alpine plants and tropical trees have been found to have low functional redundancy and are likely to support the most valuable functions in an ecosystem. And so there are no other species carrying those functions for their communities. So when the rare plants are gone, some of those functions are lost too. But also what's important to us, particularly at the Botanical Garden, and I'm sure for you all as well, is the loss of people-plant connections. People are increasingly disconnected from the natural world. And we know that native plants, plant species in general, are necessary for food security, ecosystem resilience, and emergency preparedness. At the Botanical Garden, we're having our safety and security meeting tomorrow morning at 11 a.m. in preparation for this tropical storm that we anticipate to be coming to our area this week. We know that native plants help to secure systems from natural disasters, and then having a good locally sourced native plant materials can help us prepare for uncertainty in the future. We also know that there are documented health benefits for people who spend time in nature, including increase in anti-cancer proteins, decreased decreases in blood pressure, heart rate, stress hormones, anxiety, depression, fatigue, and inflammation. Time in nature can be good medicine. And as a botanical garden, one of the best things we do is connect people with plants. And this is one of my favorite photos of a field trip in our urban spice garden. So another thing we do at the botanical garden is translate what we know about how plants grow to help people connect with them better. So this is an example of a guide that our conservation horticulturist Heather Alley prepared. And it's our guide to propagation and characteristics of our favorite Georgia natives. And it was part 1, 30 perennials for pollinators. Not only how to grow them from seed, how to propagate them from cuttings, where they grow best, who they attract. So this and other kinds of technologies that we can transfer can help increase native plants on the landscape. And I'll just take a moment here. Knowing some of you have an interest in urban settings, I can name at least 5 very rare plants that call urban communities home in Georgia. These include a rare orchid, it includes a Georgia aster, it includes a really neat carnation that I'll have a picture of in a little bit. So we're not talking about places that are really remote and wild. We're also talking about places in our cities. In fact, there was just a story on Georgia Public Broadcasting, rebroadcast this past week, about a group in Macon that were removing English ivy to create space for an endangered carnation, fringed campion. So it's really neat to think about we can have a big impact everywhere. The Southeastern United States, in particular, is interesting to me because it's been determined to be the 36th biodiversity hotspot. What makes a place a hotspot? It's high endemism and very high threat of extinction. Natural habitats that are threatened to disappear. And if you look at our rates of plant diversity, blue and green being the highest in the United States, you see that the Southeastern United States rivals the California floristic province in terms of diversity and endemism. At the State Botanical Garden, it's helpful for you to know something more about the garden. It's here on your campus. We have been coordinating the Georgia Plant Conservation Alliance for nearly 30 years. So this is a group of conservationists and agencies, federal, state, local agencies, other universities, other botanical gardens, over 100 agencies participating now to identify the highest priority needs for plants in Georgia. And this has led to significant wins in our state and across the Southeastern United States. One of the wins is the Georgia Plant Conservation Alliance has helped to establish 14 other state-based plant conservation alliances in nearby states. So to identify the priorities that are needed and then get out there and do it. Here's a map that shows where the rare plants are distributed across Georgia. They're all over Georgia, and as you might imagine, across state lines as well in the Southeastern United States. So together as an alliance, and you all can be a part of it, we're looking for students and colleagues to be a part of the Georgia Plant Conservation Alliance. We participate in regulatory oversight through our agency partners, management with the landowners, whether they be public landowners or private landowners, and then we apply research and conservation to maintain our biodiversity in our state, and as the State Botanical Garden, that's important. What I also want to point out with this map is over 90% of land in Georgia is privately owned. So if we're going to move the needle, we need to be working together in order to conserve the biodiversity of our state. And this slide illustrates some of the plants that we work with. And I'll bet you can guess which one is fringed campion, right there in the middle. It's the fringy petals. You can't get away from it. It's called pinked petals, and it's in the carnation family or the pink family. So that's one of the many that we've worked on to conserve. Right there in the middle is an example of the State Wildlife Action Plan. This is the one that was completed in 2015. Since 2005, every state in our nation has been required to prepare a state wildlife action plan. How many in the room have heard of the state wildlife action plans? Yeah, no, it's a fairly obscure thing, but I'll say when they first came out in 2005, and states were mandated to prepare a state wildlife action plan, and what this does is create a way for federal dollars to flow into the states for wildlife conservation. And only 8 states even mentioned plants in their state wildlife action plan in that first go-around. Georgia was one of 8, and we feel that it was because of the Georgia Plant Conservation Alliance, the partnerships and the trust that had been built up in previous years that led to the need to include plants. Now, again, how many of you think wildlife can be conserved without plants? I agree. Plants are necessary for life. Plants not only protect us, they are necessary for life. And so we are currently working, so these state wildlife action plans are renewed every 10 years. So this is the 2015. More states mentioned plants in their 2015 state wildlife action plan, and we are currently working with our Department of Natural Resources to update the 2025 state wildlife action plan. If you can get high priority actions for plants listed in the state wildlife action plan, then you can get federal dollars for conserving plants. Why is this important? Well, because plants make up more than 50% of the species listed under the Endangered Species Act, more than 50%, and they receive less than 5% of federal dollars for conservation. And again, if we're not conserving our plants, we're not doing a great job of conserving our wildlife either. And we received a few years ago, we're still in the process of completing this project, the first federal recovery dollars. Again, these are allocated through federal action, and they are designated for priority conservation actions listed in state wildlife action plans. Georgia, the Georgia Department of Natural Resources and the State Botanical Garden received the first dollars for recovery of rare plants that have ever been awarded. Usually it goes to animals with eyelashes, or they're kind of charismatic, but we're excited to be working with our plants too. And this slide shows examples of species that we've published peer-reviewed scientific papers on for the techniques for conserving them, either propagating them or restoring them. This is just a sample. And most of these are papers that were published in scientific journals with students as the first author through our collaborations. So important progress and impact. So my first case study is Rhus machoii. And this is an example of how we're, again, asking that question, how can we take what we know about how plants grow and apply that to how to best conserve them? Dwarf sumac is a federally endangered shrub. It has hairy legs. It's a very hairy plant. What's interesting about dwarf sumac, pictured here with its fruits, is that it has separate male and female plants. That's going to be important for my story. And so it was known, it usually grows in clonal colonies in rocky woodlands. It will grow to be about 1 to 3 feet tall, very hairy stems and leaves. It flowers from June to August, highly imperiled. Originally, it occurred from Virginia to Florida. It's now extinct in 2 states, in Florida, and I believe in South Carolina. And in Georgia, we only had 2 remaining populations. The other dots you see on the map are where it was documented from in our heritage data, but the 2 dots circled were the 2 populations known from Georgia. And also the different plants, the different male and female plants respond differently to habitat conditions. Female plants tend to stop producing flowers and fruits as shade competition takes over. So they will stop producing flowers before males will. So female plants, those that are producing flowers that can produce fruits, really need more sunny conditions. In Georgia, our 2 populations, one was male and one was female. So if I'm looking at the map, the one to the left was male on a wildlife management area, and the other one was female, pretty close to Atlanta. So this would be a plant that I would say likes urban areas. And the male population, when I visited it for the first time, this is the wildlife management area, there were only 5 stems remaining. Whether they were all the same individual, we did not know at the time. So there were only 5 stems remaining, and we collected 3 of them to bring back to the botanical garden to clonally propagate as a safeguarding effort, because we were going to apply ecological fire to the site to try to burn back the competing vegetation and make conditions in the habitat more favorable for Rus moshoii. So this is the male population. This is the female population in Covington, Georgia. Very near to Atlanta. You can see it's growing under a water tower. And so these are the interns at the time. Both of them have gone on to successful careers. And they helped me to map the female population there. So our intention was to help bring back a more natural population. And so very close to Valentine's Day in 2010, we introduced female plants to the male population, kind of a botanical dating service. And so you can see this is my graph of the wildlife management population. So in 2009, when we began documenting it, there were only a few stems, less than 10, obviously, down to 2 at one point. And then in February 2010, we introduced 22 female plants to the male population. And we started monitoring what was happening. And as we continued to monitor, 22 stems became 50 stems, 50 stems became 200 stems, until this graph stops in 2014. But the last time people went out to monitor Rus moshoii, there were over 1,000 stems there, mixed plants, producing flowers and producing fruits. So we feel like right plant, right place, right place, right plant. This was successful, that they were, but we asked ourselves, and you might be asking the same question, 1,000 stems, who cares? Maybe that's the same plant, just really being very successful. How do we know that this really truly is a success? Well, what we might call a success is to say there are genetically distinct individuals in the population, right? How many are in the population? So we looked back to the literature, and the previous genetic studies had been done in the early 90s. Actually, my PhD advisor was part of this study. And they sampled populations from North Carolina and Virginia, the 2 other states that have largish populations of Rus moshoii. You'll notice they're smaller than what we ended up with here in Georgia. 280 to 500 stems in these populations. They were occurring on DoD installations. Does anyone know why that might be? They burn things on DoD installations. They drop bombs, and then there's lots of fire, and that's good for Rus moshoii. So anyway, there were some pretty stable populations of Rus moshoii in North Carolina and Virginia, and what they found is that there were, on average, 4.1 distinct genotypes on average in these 2 populations, right? So anywhere between 1 multilocus genotype to 7 multilocus genotypes per population, but on average 4 distinct genetic individuals. When we looked at our Georgia populations, we found in the Newton County Covington females, we found 6 genetically distinct individuals in a population with about 150 stems. That's pretty good for Georgia. We feel pretty good about that. And then when we went to study the male population, again, the one that had recovered up from only 2 stems, we found 10 distinct genetic individuals, at least, at a minimum. That meant something was going on underground. It was waiting for conservation. It was waiting for fire to hit the landscape. And for me, it's really an inspirational story. It means do not give up on a site. Don't give up on a plant. We only had 2 stems. Now we have over 1,000, and we have more distinct genotypes than were found previously in populations. And when we moved those 22 individuals over to the wildlife management area, we actually were doing it carefully, thinking we were making good decisions, and we actually captured 5 of those 6 genotypes when we moved it over. Okay, so then more horticulture. What do we know about the seeds? We did seed germination trials with our colleagues at Georgia Tech. This is a slice of a cross-section of what the seed looks like on the far left. It's a very thick seed coat. likely distributed by an animal, needing to pass through the gut of an animal more likely than not. When we tried trials with a congeneric species, a very common Rhus glabra, smooth sumac, we found that we needed to drop the seeds into hydrochloric acid for 6 hours. That's a really long scarification time, but these seeds are pretty tough, and that's what they required in order to germinate. Then we requested seed from a seed bank. A seed bank is something, as you know, that's out there to save seed for the future. It's a safeguarding mechanism. So seeds were being held of Rhus moshoii in this seed bank. They were collected in 2005, and we requested, I think, how many did we request? Let me see if I have it in my notes. We requested 148 seeds. When we tested the seed, only, or 93% of the seed that had been held, resources put towards saving them, 93% of them were empty. 3 of these seeds germinated. Others were damaged from the scarification process or contaminated, and the resulting seedlings did not survive the initial tests for germination. So that's not a great outcome. We had to try harder. So we now had a well-established population of Rhus moshoii here in Georgia, which was producing seed. So we collected 114 seeds from the population in Georgia, and only of 114, only 2 were not contaminated and germinated. And so they survived in the greenhouse for a year and outside for another year. So these are 2 of the survivors out of 114 seed. Again, not great odds for this plant. We're learning something about the plant. We've learned something about its habitat requirements. We've learned something about how it will respond to restoration, and we've learned something about its seeds. We repeated the seedling trials again with seeds. This collected from Georgia. 515 seeds were collected from females located very near to the male plants in the wildlife management area. And 243 seeds were collected from females located a little bit away from the male plants. So close, but in the same population, but right, not adjacent to them. Of the 515 seeds that were located near male plants, only 10% of them were filled, and 11 seedlings survived in soil after a month. So these are not great odds, and even worse if you're not adjacent to the potential reproducing male plant, where 243 females located from plants, away from plants, only 0.4% of the seeds were filled, and 0 seedlings survived. So, and again, we collected another 2,600 seeds from mixed populations. These were XC2 plants growing on a rooftop at Zoo Atlanta, where a population had been restored, and only 0.3% were filled and 1 seedling survived. So what do we know? What have we learned? That these plants are not producing very viable seed. We have more research to do with the biology of this seed to learn how to better propagate it. And that even if we put hundreds of seed into a seed bank, which is out there to protect species and safeguard them, that's not a guarantee, that's not a safeguard. But we've also learned that we can clonally propagate these plants, that we can grow them and mix them, and that we can begin to meet our goals for long-term storage and propagation. And they're all endangered species. All species listed under the Endangered Species Act have recovery plans. And so this helped us move a little bit closer to creating those plans. So I'm going to give you another case study, but I'm going to change the question. What can we learn about how plants grow to determine why some plants are rare and others are more common? Why are some plants rare and others very common? We were curious. So we studied a plant named Pityopsis ruthii, or Ruth's golden aster. This is a very poor competitor, at least we thought. It's found along the Ocoee and Hiawassee River. So if you like to paddle on the rivers, you might be familiar with this area in Tennessee. and it grows in the crevices of rocks. And so we thought, well, this must be a terrible competitor. There must be something weird about this plant, why it's only found in this very specific location. But as we ask this question, why are some plants rare and others more common, we can look back to a descriptive framework developed 40 years ago by Deborah Rabinowitz. This helps us to describe, because not all plants are rare for the same reason, or at least we can begin to classify this. And so this is after Deborah Rabinowitz and after Shipman et al. 2006, where the framework describes geographic distribution of plants, whether it's wide or narrow, habitat specificity, whether it's broad or restricted, and then local population size, whether it's somewhere large or everywhere small. Now, only those plants that have a wide geographic distribution, broad habitat specificity, and somewhere are large populations are the least conservation concern. All of the others are considered rare on some level and in need of some type of conservation action. And the one we're going to talk about today in this next case study has a very narrow distribution, very restricted habitat, and in some cases can have large populations, and I'm not going to be able to describe them. They can have maybe 50 to 100 individuals, which might be considered large. So it's somewhere at the bottom there, very susceptible to extinction. But we first wanted to ask the question, what do we know? What has the literature told us about why some plants are rare and others more common? So we did a meta-analysis. We looked at 153 peer-reviewed articles published between 1978 and 2020. And these are articles that did comparisons between common and rare species. So within the same study, that was a requirement. The meta-analysis included 252 rare species and 267 common species. You can see what we excluded in our meta-analysis. It was phylogenetically controlled. I'll show you some of those results. And we were looking for eco-evolutionary causes for rarity. What could we understand about the ecology and the evolutionary background of these species that might give us clues for why a plant might be rare and another one more common? We included parameters around population genetic diversity and fitness and functional traits. And this is because we wanted to ask similar questions with Ruth's golden aster when we did our own research. So this is an example of some of the parameters we would look at for genetic diversity. How many of the loci or the locations that we were sampling of the genotype were polymorphic or had more than one allele? How many alleles were there for these loci that were polymorphic? Observed heterozygosity is a measure of genetic diversity. FST is a measure of diversity among populations, and FIS is diversity or within populations. And what we did is use Hedges G, which is a standardized mean difference comparing and contrasting parameters for rare and common species. So if the data fell below the 0, it meant that the rare species was lower than the common congener for that parameter. Maybe it had lower genetic diversity or lower fitness. So when we included population genetic metrics in our comparisons, we found of 31 studies that included natural populations, this was 37 rare and 37 common species, 31 genera, 20 different families, that in 4 cases, 4 different measures of genetic diversity, the rare plant had less genetic diversity than the common plant. So what we found is that by this measure, rare plants may have less evolutionary potential to adapt to change or to adapt to the conditions in front of them. This is the distribution of those species included in the genetic diversity analysis, and you can see we had different types of rarity also categorized, and so across the phylogeny of those plants included in the study, the types of rarity were fairly well distributed. And that when we looked at where the studies occurred for the types of rarity included in our meta-analysis, they were fairly evenly distributed across the globe, although you can see in those parts of the world where there are no dots, it means we're data deficient, and we need to do a better job of studying those plants in those areas. Looking at fitness components, which might include juvenile recruitment, survival, growth, and fitness, which can be measured in multiple ways. 2 of those fitness components were found to be significantly lower for rare plants when compared to common congeners. They're relatives, they're cousins. When I say congener, it means they're in the same genus, but they're not the same species. Okay, good. And then when we look at functional traits, one of the ones that was significantly lower for rare plants was reproductive size, or the size of the reproductive organs. And we looked at pollination measures. We found significantly lower success with self-pollination in the published literature, and significantly lower success with hand pollination. So just to kind of bring it all home, we found through the meta-analysis that rare species may have limited ability to adapt to environmental change if neutral genetic variation is representative of fitness-related genetic variation. We found that rare plants had significantly lower survival in certain cases with certain studies. Rare species had smaller reproductive traits but no additional signature for rarity and functional traits. And maybe there's a reason to believe that rare plants could have less efficient pollination. And both this meta-analysis and the research I presented on Rhus machoii indicate that there may be something going on with seed biology, seed pollination, because we were finding very low filled seed in Rhus machoii. So to move on to the case study with Ruth's golden aster, we compared it to the very common silky golden aster, or Pityopsis graminifolia. So again, these are congeners or cousins. They're in the same genus, they're just different, two different species. Pityopsis ruthii is very, very rare, and Pityopsis graminifolia is quite common. But before we get to the description of these 2 species, let's think about the ways a plant has, usually rooted in the ground, to respond to a changing environment. They can get the heck out. They can migrate. They can adapt. You know, if you have genetic diversity with directed selection, you could change over time in a way that might make you better suited to the new environment. Or you can acclimate, and that means you could be plastic. You could be able to show different functional traits or different types of growth in different environments. So in a dry environment, you may put more into producing seeds. In a very wet environment, you put more effort or resources into producing leaves for growth. So acclimation is a way of describing plasticity in plants, and it's one of the ways that plants have for responding to a changing environment. And plants can be quite plastic. They have that characteristic. But it's also recognized that not all plants have the same abilities to be plastic. So that was part of our question. And then the final, not so happy outcome could be you would go extinct. You would just not be able to respond well to a changing environment and eventually blink out of the landscape, which we've been talking about for a while, and it's something we want to avoid. So genetic diversity measures can give us a clue as to whether a species has potential to adapt, and phenotypic plasticity gives us an idea of the species' potential to acclimate. So here are the 2 species we were looking at. Pityopsis ruthii is considered susceptible to extinction. It's listed as an endangered species. Whereas Pityopsis graminifolia is found nearly everywhere. You've seen it. It's flowering right now. It's flowering on roadsides and in fields and in forests right now. And it's of least conservation concern. If we look at their distribution maps, I had to circle the one little teeny tiny dot for Pityopsis ruthii because it's really only along 2 rivers in Tennessee. Just a few populations. And whereas Pityopsis graminifolia is found throughout the southeastern United States, and for our research, we sampled across the range. So one of the first things we learned in our study is that we could grow both the rare and the common plant. And in fact, many times in our research, we find the rare plant is easier to germinate and grow. I do not have an answer for that. So this is our team of conservation interns, graduate students, and our research associate, Will Rogers, growing the plants in the greenhouse. And we took some of the leaves and did a population genetics study. And again, we found that by some measures, including Ney's genetic diversity, and the number of alleles per locus, and the overall MANOVA, which is a test of the different measures of genetic diversity, was significantly lower for our rare plant compared to our common plant. Not unexpected. We just sort of verified what we might have expected. But again, it means that their genetic diversity could limit the ability of our rare species to adapt to a changing environment. And then we did growth chamber studies to study plasticity. So we took a parent plant and we cloned it and we grew it under ambient conditions in a growth chamber, and then we changed light, temperature, and water. And then we measured a number of different traits in these species. So we looked at height of the plants, number of leaves, buds and flowers. They didn't flower well in the growth chambers. Root length, shoot mass, root mass, total mass, specific leaf area, root shoot ratio, root mass fraction, and shoot to root levels. But all of these are measures of how the plant is putting its resources, its limited resources, into growth, whether it's putting it into roots or into leaves or producing flowers for the next generation. And so these are the response curves. These are examples of response curves that can be mapped for a changing environment. So along the bottom axis, the environment will change. If the trait values do not change, for example, the one on the far left, there's no evidence for plasticity. If with a changing environment, the same clone is the trait that we're measuring changes with a different environment, there is some evidence for plasticity. And if there's significant differences with environmental change and when we're measuring it in the growth chamber for a trait, we have evidence for highly variable. So we did statistical analysis and looked at the response curves, and I'm just going to present the response curves for certain traits to you now. So when we looked at reduced light, this might be an example of how competition is moving in. Trees are moving into an area where these plants are grown, which is known to occur in nature. We found that for the rare plant, there was evidence for plasticity. This is a plant that changed the way it grew when there was less light. Whereas with the common plant, we didn't see evidence for plasticity when light changed. For changes in the amount of water, and Pityopsis ruthii grows in a place where it floods regularly, we found evidence for plasticity in the rare plant when there's ambient amounts of water and increased water. Whereas with the common plant, the graphs on the right, we found no strong evidence for plasticity. So again, the rare plant in this case showed a plastic response to increased water. We didn't think that there would be a significant plastic response in the rare plant, but we did find it in this case. And then finally, looking at temperature, we did not find a plastic response for changes or an increase in temperature for the rare plant, but we did see a plastic response for the more common one. We found this quite interesting. And the reason is that the kind of plasticity in our rare plant could be facilitating its very narrow habitat specialization. So remember, one of those factors for rarity. So Pityopsis ruthii's ability to adapt to changing light or competition or increased water through flooding may be allowing it to hold on into these narrow crevices in the rocks along the Hiawassee and Ocoee River. It is only found in 4 populations along the Hiawassee and 3 populations around the Ocoee, and nowhere else in the world. So that may be allowing it to stay in that very narrow distribution. However, it showed very limited plasticity or ability to grow, and it showed limited genetic diversity compared to the common congener. Our hypothesis is that its limited plasticity in terms of increased temperature could be limiting its ability to get out of that river basin along the Hiawassee and Ocoee rivers. There are significant mountains around this. The east-west running rivers could be limiting the ability of this species to survive and distribute outside of the river basins where it is found. So we found this quite interesting because it gives us a clue. It helps us understand that framework for why certain plants are rare and others are more common. And these can be applied in a variety of environments, in a variety of situations. We can begin to apply them too when we model the trajectories for species in natural areas and in urban areas. So I want to thank my funders. I want to thank my collaborators. We did receive significant funding from National Science Foundation. We did participate and collaborate with the U.S. Fish and Wildlife Service and the Tennessee Valley Authority. So I want to leave you with what we've learned. And I want to recognize Will Rogers, who's our research associate at the State Botanical Garden, who's really can grow anything. He could root a pencil. He can go out in the field, and he can find the plants in the field, and then he can bring them back to the lab and study them for their genetic diversity. So what we've learned that best practices in conservation horticulture rely on continued understanding and observations in nature. Can a seed be stored? How to grow it in the nursery or in the greenhouse? What soil matrix to use? By making careful observations about how plants respond to changing habitat conditions, we learn much about their life history and biology and constraints and responses to environmental change. Pityopsis was a good example from this. And through the application of conservation horticulture and translation of our horticultural research, we are training a new generation of conservationists and addressing the conservation needs of hundreds of plants in the southeastern United States. Thank you.